Cas9 technology, do a gene repair, and then try to transplant back these cells to the patients. So the requirements of these artificials definitely are different for these different applications, and maybe if the industry that is focused on developing these products can, you know, develop different areas or different characteristics of these artificial BMEs depending on the application will be important. For example, we are aware that in the example of regenerative medicine, still some researchers are expanding the organoid cells in animal-derived BME because the expansion capacity is still an advantage. But in order to be able to do the step of the transplantation, then in the final 1 or 2 passages, the cells are transferred into an artificial BME in order to be able to do the switch. And definitely one of the challenges that these that the industry that will develop these artificial BMEs, or the academic labs that are working and putting efforts on this, is that they need to address definitely the size production because, I mean, already now we are facing, especially post-pandemia, still some issues getting access to the amount of animal-derived material that we need. But as we were showing in this estimation of the market of use of organoids, and you know, this is just some numbers, but indicates the constant growth, that whatever artificial BME can be generated, can be produced in a large scale is definitely a must. Otherwise, there is a limitation on being able to implement these new artificial BMEs in everyday work. And definitely, and associated to this is the cost, because already culturing 3D cultures already have more costs than culturing 2D cell conventional 2D cell lines. And of course now, because they are probably prototypes and very specialized, artificial BMEs are relatively expensive, so it will be necessary that the cost is reduced to also be able to implement this alternative to the animal-derived BMEs. So with this, I'm finishing my presentation. I was just again trying from a more industry perspective, first present, you know, how we are using these 3D cultures and how much we are depending on these animal-derived the BMEs, and what will be the important aspects to tackle in order to be able to transfer to using the artificial BMEs. So thank you for your attention, and I'll be happy to address any question from the audience.
Thank you very much, Sylvia, for this nice update. If there are questions, please write them down in the Q&A in the bottom of the screen. So far, there are no questions yet from the audience, but maybe I can kick off with one question to you, Sylvia. If I understood, well, actually I have two, but the first one is, if I have understood you correctly, then you will use animal BMEs in the preclinical phase, for instance, and then develop your product. And then when you are going to personalized medicine, you will change to artificial. That's the idea, right?
Well, definitely this is a required step for when you want to do regenerative medicine application. In terms of diagnostics, we are already exploring with authorities like EMA how, you know, in this, there is this IVDR now regulation in order to perform this in vitro test. So to evaluate how much is required to perform this diagnostic in a very well-defined conditions, including what type of ECM we are using, or if the results, at least for diagnostic, could still be performed in these less controlled BMEs.
But do you expect a loss of performance when you?
It's definitely something that needs to be evaluated. So, for example, this example of so far all the validations in terms of predictability of the organoids has been performed in these animal-derived BMEs. There are several publications and also our own work. So far the animal-derived maintains this characteristic. If we will have to change to ECMs, artificial ECMs, definitely this needs to be confirmed, and if we are not getting results, then it will be very important to join efforts with the experts developing these artificial ECMs, what type of characteristics we are missing that we cannot reproduce, for example, this clinical outcome.
Okay, thank you. In the meantime, we have some questions here. It's from Kathy Mary. Thank you for a fantastic talk. How important do you find the mechanical properties of the gels are compared to their functionalization?
Definitely, this is a very interesting question, and it's definitely one of the challenges that we are facing, because as an organoid company, we are not, our knowledge about all these physical characteristics of a strength and force that we learn when we read all these publications about the artificial BMEs, how important they are and how much they can indeed modify the cellular response. It's a very important question that, and it's also a bit complex, so we are now, let's say, having a bit of an easy approach that as soon as organoids are able to reproduce, in our case, patient response or maintain the same heterogenic mutation landscape that we identify on the in the original tissue, that we don't put that much effort. So let's say we are trying to identify for what type of applications, drug response or representing the available ECMs are suitable, and if for certain diseases or properties we see that what it's available is not representing the in vivo situation, then definitely we will put more effort to understand if we need to work with this more artificial that allows all these modifications on the physical characteristics of the ECMs.
Okay, thank you. And then Simona asks, could you expand a bit on the diagnostic tests with cancer organoids and challenges observed with using BMEs?
So one of the challenge, right now these diagnostics in cancer are done by seeding the organoids in a 384 well plate with certain percentage of matrigel and doing a cell viability readout. We had to modify the way of seeding the organoids with the matrigel because the viscosity and the had some impact on the readouts that we were using to evaluate the viability. We are also exploring imaging readouts, and there also the fact that the organoids are on 3D and sometimes they are not on the same Z-stack are having some impact. So we are working on trying to improve the readouts and the applications of the diagnostic. But as I say, we are taking a bit of an approach that whatever, for example, for basic chemotherapies like 5-FU, oxaliplatin, irinotecan, we see that the current setup of doing the diagnostic with the semi-suspension situation with ECM is good enough to reproduce the patient response.
Okay. Then there is maybe time for one more question here. What should be done technically to test the effect of a certain treatment on the differentiation of intestinal epithelial cells, but to do so using already differentiated organoids?
So can you maybe repeat?
Yeah, it's a bit, I also find it difficult to grab actually the question. But what should be done technically to test the effect of a certain treatment on the differentiation of intestinal epithelial cells? Doing this by using already differentiated organoids. I think that's the point of the question.
Well, I don't know if the question refers to healthy differentiated cells or disease, because in principle, we are not testing, I'm not sure if I am understanding the question, because we are not, we have identified the growth conditions to either expand epithelial cells or to enrich for the different differentiated cells that you can have. So if by this is the person is asking if using Notch, what type of Notch inhibitor or whatever will improve the differentiation state of organoids, this is something that we already are able to do. So I'm not sure about the technical questions, but I guess I'm not understanding correctly the question. Sorry about that.
Yeah, maybe they can contact you by email.
Sure, definitely. Yeah.
and C3Rs maybe can arrange that. Yeah, I think Hakan Berg Aydin says thank you for the cost effectiveness and controllability of the BMEs. Decellularized or totally synthetic approaches will be favorable in time, ? So he wants to know if we have to move towards a completely synthetic BMEs.
Well, I mean, from a perspective that, of course, we would like to not need the use of animals to perform our research. I mean, we already felt that using organoids, we could have an impact on the use of animals, right? Because of, for example, the example we gave about the toxicology. We are really aiming that we can replace animal experiments in toxicology by using organoids derived from few of them, and definitely human. That is the predictability that we need. So in the same range, we would love to not depend on animal-derived BMEs to perform our activity, the expansion of the organoids. But at the same time, of course, there is also a need to develop better drugs and new drugs for human patients. So in the meanwhile, we need to use these animal-derived BMEs because, going back to the cost-effective, and maybe as a summary of the discussion at the end of the presentation, we need artificial BMEs that allowed a proper expansion of the cells in a timely manner and definitely in a cost-reduced manner compared to what is available now. That, of course, there are some products already, but in terms of high implementation of the expansion of these models, it still is quite expensive and not reaching the level of expansion that we have now with the other models.
Okay, thank you. I think we have to move on. So I suggest that those who had a question that was not addressed to contact Sylvia directly.
Sure.
Or maybe repeat it when it's obvious for any of the two other presentations. So, okay, thank you very much, Sylvia.
Thank you, Erwin.
So let's move on to the next speaker. That's Jakob Benedict Seidelin. He's associate research professor in gastroenterology at University of Copenhagen in Herlev Hospital. He's also consultant in gastroenterology with focus on inflammatory bowel disease, and he's head of the clinical research unit. His main research interests are wound healing mechanisms of the intestine and regenerative medicine, as well as clinical trials in inflammatory bowel disease. He was also visiting scientist at the Langer Laboratory, Koch Institute, MIT, Cambridge, Massachusetts, doing research in regenerative mechanisms in this area. So Jakob, the floor is yours.
Thank you very much, and thank you for inviting me to this webinar. So this is going to be a much more focused presentation on a specific disease, and in that way somewhat different from what you have learned or seen previously at the webinar. So I will just walk you through the disease that we are going to talk about. Just a second. This is inflammatory bowel disease, and specifically ulcerative colitis, as you see in the right panel here. It is an inflammatory disease affecting the colon, and if it becomes acute severe ulcerative colitis, this superficial inflammation of the mucosa will typically involve the whole colon. So this is what it looks like at endoscopy. This is a picture from an, I'm sorry, just a second. This is a picture from a normal colon, and this is varying degrees of inflammation. And as you can see here, this is the severe form of ulcerative colitis. This is relatively common. It affects approximately 20% of the patients and is typically seen among younger patients. So the current treatment for this is admission to a hospital ward, and then typically, not typically, but always IV high-dose glucocorticoids in the form of prednisolone. And if this doesn't work, rescue anti-TNF treatment in the form of infliximab, and then surgery where the patient is colectomized if the anti-TNF does not have an effect, or the combination of glucocorticoids and anti-TNF is ineffective. So as you can see, there are some unmet needs on each of these steps. Approximately 40% of the patients are unresponsive to corticosteroids. Of those, approximately 40% again would be unresponsive to anti-TNFs. And this results in a substantial number, that is approximately 15% of the patients with acute severe ulcerative colitis needing colectomy. This is still a type of surgery which carries a risk of severe complications. And so apart from the patients losing their colon, there might be several reasons to look for a better treatment of the patients. So you may not, I mean, it is obvious that this intestine is the barrier function of this intestine that you see here is completely abolished, and this was shown in one classic example more than 20 years ago by measuring the conductance over the epithelium, which is increased due to the loss of epithelium. But also, if you do look at the genetic or GWAS studies, barrier function and restitution plays an important role in IBD, among a lot of other factors, of course. So I think regeneration is as a target. There are some studies out there showing that this might be potentially a way to go. I think a classic study is this study where they treated ulcerative colitis with enemas with EGF, and it was actually efficient in treating the patients. So this is a non-anti-inflammatory treatment of IBD, targeting regeneration. Another line of evidence are the many different studies that during the past 10 years have been performed, showing that in mice that you can induce experimental colitis and then treat the colitis with introids to induce healing of the mucosa. And then I think another very important finding was recently shown by the Roggen group, showing that if you take out organoids from patients with active ulcerative colitis, then immediately after taking the organoids out, they will retain the inflammatory quality or dysfunction of the epithelium. But if you grow them for one passage or more, they lose this inflammatory way of working. So, and then they become you can't tell them from normal organoids at that point. So we set out to discuss the possible the obstacles and the possible ways that using organoids as a regenerative therapy, how that could be done in IBD in this paper in Molecular Medicine in 2017. And of course discussed most importantly the preclinical phase type of studies that need to be done to achieve this. And so we set out some questions that we thought we need data on to be able to move on to perhaps a human model. And this was most importantly a way to harvest and establish organoids from freshly isolated epithelial cells and to expand those cells under GMP conditions. Then it is of course some sort of a delivery method. I think we would need to have a suitable model that could be used in a proof of concept design or a early phase 1 design, and then a method to trace delivered cells to prove embedding of the cells. And we've been discussing pros and cons of Matrigel, especially Mona Risi discussed this in his talk, and it has also been discussed by Sylvia just now. We think that the main obstacles from using this in clinical condition is the undefined nature of Matrigel. Also that it is based on a cancer cell line. We know that there is batch-to-batch variability, and it might not be scalable in an economical fashion. And if we need to modify the matrix, it is somewhat difficult. So, and it has also been covered the advantages of synthetic gels, and this was especially covered by Simone Risi in his talk. But we thought at that point that there was a limited information on the long-term efficiency of these synthetic defined gels in stem cell maintenance and expansion, and also differentiation capacity, and de novo establishment of organoids. And therefore we set out to study this in collaboration with Simone Rizzi's group in Switzerland. So at first we screened a lot of different gels, and we ended up with this CN99 gel, which is a PEG-based, fully defined PEG-based hydrogel, which has a stiffness that varies over time, becoming quite similar to Matrigel over time when we grow the cells. And what we could see from that was that we were indeed able to establish the organoid cultures directly in this gel without a pre-amplification of cells in Matrigel, which has been used previously. We could establish the cells directly in CN99. Although, just a second, with a lower efficiency than with Matrigel, as you see here. But when we came to expanding the organoids at following passages, we saw with this gel, we saw an identical incremental factor of cells using the CN99 gel. And also, and we could use that to simply calculate if this method would be suitable for within a few passages to reach a sufficient number of cells comparable to the number of cells used in the mice studies to do sort of a small scale study in humans, and found that within 2 passages we would be able to have a sufficient number of cells for such a purpose. Then I think it is important to think of the cells as a product that needs a lot of quality assessment before it can be used for human use. And we did, actually our pathologist, when she saw the organoids, she was very concerned because they look more like carcinoma cells being grown in this very stem cell stimulating medium that normal stem cell medium is. But if you differentiate the cells using the CN99 gel and Metrogel, you get differentiation very similar to what you see in the human colon. And more importantly, if we did RNA-seq of our cells, we could see that they cluster very closely together with biopsies from healthy controls and very far away from cancer cells. And that's one aspect, but we also showed that the cells did not acquire any nucleotide substitutions at passage 10. At passage 10 we did that experiment, and we didn't see any genetic instability in this culture system. This goes both for Metrogel and CN99. So then a little bit of thoughts on the culture medium. We have been trying to humanize the culture medium in other experiments that I will not show you, but a simple way to deal with this could be simply to dilute out the growth factors, and this was one of those experiments where a simple washing quite quickly take away the important growth factors that could be a problem in human applications from the cell product. Then just to briefly mention our thoughts on how you could see if you really have embedding of the epithelial cells. We did a study where we wanted to see if we by endoscope could trace these cells, and so we have what is called a CLE, which is a confocal laser endo microscope. This is the endoscope. It's a normal endoscope for human use, and this is, by the way, Frederick Bergenheim, who was our PhD student and has been doing a lot of the lab work on the studies I mentioned for you. And this is how an organoid looks by using this endoscope. And so we think it is unlikely that we're going to use genetic modified cells for this purpose. So we screened a lot of different fluorophores that could be usable for clinical applications, and by different qualities that we wanted the fluorophores to have, we could exclude many of them and ended up with a fluorescein-based coloring that was detectable by CLE, and which could be used in a experiment with mice where we took TMG mice and took out their epithelial cells and grew organoids, and then stained them with this, and ex vivo were able to detect embedding of the cells with the endoscope, as I just showed you. So I think briefly here at last, I want to just share some thoughts on how this could be advanced into maybe a clinical trial, and then I think it is interesting to have some model studies from other areas, and I like this study a lot, a Lancet paper from 2013, where they took out adipose fat grafts and grew them ex vivo and transplanted them back to healthy subjects. This was a small-scale experiment, and I think this is both in terms of patients going into the experiment, but also in terms of the way the transplantation was done. It was done in the forearm of the patients, and they could here with MRI follow how this transplant performed compared to another way of transplanting the cells. So I think it should be considered to use a small-scale model, and I'm not saying that the particular model I show you here, which is a model we've been working on, is the way to go forward, but this is just an example of a small-scale inflammation model of the intestine that might be usable for the very initial proof of concept studies. So I wish to thank, of course, Frederik Bergenheim, who did a lot of the experimental work that you have seen here, and Ole Hohen Nielsen, who is our professor at our lab, and also Simone Rissi, and Julia Fregni, and the whole group in Switzerland who helped with the matrix studies, and Jeff Cobb from Brigham and Women's Hospital, who has also been involved in these studies, as well as Kim Jensen, who was involved in the mice studies. I think that's it.
Thank you very much, Jakob. So if you have questions, please write them down in the Q&A. There is one question at this point from Christopher, and he says, you mentioned the cost of synthetic BMEs is a limitation, and this may refrain researchers from shifting to non-animal based BMEs. That's the risk, of course. Is there any evidence that reduced variability when compared to animal-based BMEs in, for instance, treatment responses and reproducibility can compensate for this limitation? In other words, in the end, it would cost the same. It's a long question, but it's about the price.
Yes, so I think the real question is if the matrigel is so efficient that that you could perhaps avoid what would be more efficient in large-scale experiments.
Yeah, that the advantages override the extra costs. Yeah.
I actually think that this CN99 gel shows that we are quite close at getting to a point where synthetic gels might be as efficient as cell-based gels. And I think the scalability is, I mean, you can see now with the problems of providing matrigel at the moment, and the problem they had by freezing or closing down the production and starting it up again in Corning. I think that this shows some of the problems using this in large-scale applications. I think also from a approval point of view that the synthetic gels have many advantages, and that some of the limitations are narrowed down at the moment.
Okay. I cannot see any questions from, oh, there is one popping up. I should have. From Omar Shamhat, what are the risks of intestinal stem cells or organoid transplantation in terms of giving rise to colon cancer? Or the relevant studies in animals? I mean, all it takes for it to lead to cancer is to have one transformed cell in the transplanted material.
Yes. So I agree completely. This is one of the main issues with this technique, and I think the expansion of the introids in this very with a lot of potent growth factors could impose mutations or genetic instability. So we didn't see that in our experiments. We tested, of course, not a sufficient number. It was 6 different patients that were tested at passage 10 with this in this particular. But it has been done also in other by other groups, showing similar results. I think one important thing there is that it would make it hard to do transplantation from another person to a patient. I often get the question, why transplant the sick cells back to the sick patient? But I think that would limit some of the risk of neoplasia using this method. But I agree completely that this is one of the major issues that need to be addressed. And I think if one were to do small-scale experiments, we could actually, as they did in this model study, where they took out by surgery the transplanted area, this could be done a few weeks after the transplantation if you wanted to do such for security reasons. Or I think that would be manageable.
Thank you, Jakob. Then there was another question from Christopher. What regulatory constraints do you face when performing such trials in humans?
So we are not there at all, but we thought that one way to go would be to start an advisory track at the Medicines Agency, and we actually just when Corona started, we filed in for that, just to explore what kind of additional experiments they would like to have at this point. And I think that was at least our way of thinking of going forward with this endeavor. But that is not to say that we are in any way near any human experiment at our group. So I don't want to make the impression that is what we're working on. But we had communication, but it halted due to this Corona situation.
Okay, so there are no further questions from the audience. I have still one question for you, Jakob. I still have 2 minutes. It's a bit inspired by questions from the first round of question and answers, and it's about the heterogeneity of the heterogeneity of the human organoids. You take biopsies from a patient. This is not one single cell type you collect. You have whatever is present at that site in the intestine. Then you transform that in organoids in this artificial BMI, for instance. Are all these cells still there when you have your mature organoid, or is there a selection at cellular level? Do you know that?
So I think when we look into the transcriptomics studies, it looks very similar to colonic epithelium. So that's one thing. I think you would need to do single cell sequencing if you really wanted to show if that's the case. But the way I see it, and when you put these cells into differentiation medium, you get a quite you get all the sorry, you get all the different cell lineage markers that we tested for, at least in those experiments.
And that's representative for the intestine?
Yes.
Yeah. Okay. Okay. We'll leave it there. Thank you very much, Jakob.
Thank you.
We move on to the next speaker, and that is Christian Tryggvason. He is he's founder of the company Biolamina, which is based actually on the research work of his father at Karolinska Institute. The vision of Biolamina is to make it simpler to grow primary cells, including stem cells and any other type of mature cells. And now, after 12 years as CEO, he focuses on solving cell culture problems in different areas, like helping develop better cell culture models for drug development. Christian, the floor is yours.
Thank you. Do you see the slides?
Yes.
Great. Yeah, so I'm just going to give a short introduction.
It's not in presentation mode.
That's strange, because I see it in presentation mode. Now?
No.
What can you see?
I see the slides, but also the list of slides on the side.
Are the slides big enough? Because I don't know how to. Oh, here, use slideshow now.
Yes, that's great. That's wonderful. Thanks.
Okay. Yeah. So I'm first just going to give a very, very short introduction to Biolamina. Then I'm going to talk about the laminins, which is one of the proteins in the extracellular matrix. I will show you some examples of how these have been used mostly in the cell therapy field. And then I will basically show why they can also be used in cell therapy. So there's quite a few companies and labs that are using these in cell therapies. And then I will also show one of our recent products, which is it's a biosilk. It's a 3D culture model that can be used basically for culturing organoids and 3D cells. So Biolamina was founded in 2009. My father approached me and asked if I wanted to start a company with him, and he wanted to build a cell therapy company, but I didn't think that we had enough data to do that. So instead I proposed that let's start selling these laminins to everybody around the world, and somebody will take them into cell therapies. And so that's what we started, and in the beginning we only had a couple of products. But in 2009 2014, we actually, or not we, but Sergey Roggen and et al. They published this article how they showed like how laminin 521 could actually be used to culture embryonic stem cells in a really good way. A year after that, we made a deal with Roche, which then didn't come out anything, but they started putting some pressure on us in the quality side of things, because they realized that these could be used in therapies, and we had to adapt our processes so that today, actually in 2021, we have 5 different customers that are in cell therapy trials using our products, and that I will talk a bit more about that, qualifying our processes and so on as well. So the vision is to become the leading provider of solutions for primary cell culture to enable life-saving therapies. And this basically means either towards helping making cell therapies or helping make tools that you can use to develop therapies, basically drug discovery tools or something like that. Today I will mostly talk about how scientists, I mean, in research labs, how they basically started with some kind of cell culture problem, which they normally did in 2D, and then how they solved that problem, and then how they have actually continued with this method that they developed into cell therapy trials, and where you also need to scale up your processes. And basically, I hope that what I will show you will be interesting for you to actually use in the drug discovery process as well, and making better cell culture models, which today we haven't actually approached these kind of scientists that much, actually, because we are not a very big company, so we have been focusing mostly on these cell therapy customers. But now we have a really nice 3D cell culture system as well that can be of use in that area. So why laminin-based cell culture reagents? I mean, this is something that has already been partly shown earlier, but if we just kind of recap, basically almost all cells except circulating cells are next to a extracellular matrix. And if you look at the extracellular matrix, basically the laminins, let's see, how can you make a laser pointer? Yeah. So I mean, if we look at the closest, so the cell is here underneath, and then the laminins are the ones that are most abundant closest to the cells, and they actually bind to the cells with the integrin receptors. And then there's other proteins there, fibronectin, collagen, elastin, and these kind of things. But these are actually ones that actually interact with the integrins, and they give signals to the cells as well. So these laminins are the major component of the basement membrane in vivo. They consist of 3 different chains. There's the alpha chain, the beta chain, and the gamma chain. And there's 5 alpha chains, 3 beta chains, and 3 gamma chains. These are the only proteins in the extracellular matrix that are really tissue specifically distributed. So the heart cells have one type of laminin close to it, and let's say some dopaminergic cells have another type of laminin. And these are basically impossible to purify from tissues in native form. And if you think about major gel is actually mostly laminin 111, but it's from a mouse tumor, and it's if you run the laminin or the major gel on a gel, you could see that it's very much degraded because it's very difficult to purify these. So, and one other thing is that there's really 16 different laminins, and these have different functions. We can see that like laminin 521 is excellent for expanding pluripotent stem cells, but you put it on laminin 421, the cells will die, and so on. And basically the whole idea is that if you are working with a certain type of cell, like let's say a muscle cell, you should use laminin 211, and if you're using some other type of cell, you should actually use these, because these are the cells that are actually in the body, or these are the proteins that are next to cells in the body, and therefore if you put these on a Petri dish, they will feel like home. We see from the studies that I actually will show you a bit later on is that the reason why, I mean, this is actually a bit of an answer to Christopher's questions just in the end, is that if you use these laminins, you will actually get better, more homogeneous cells, and you can get there more easily. And basically, in theory, you could save some money because you have to repeat your, I mean, if you're doing experiments, you need to repeat them less. And if you're doing some manufacturing, you will get to the endpoint more often. So basically, the laminins are no engineered products, but these are just biorelevant proteins that are next to the cells in the body, and these therefore they work really well on a Petri dish or whatever system you want to cultivate your cells on. And it actually makes cell handling very, very easy. In the embryonic stem cell case, basically you could take I mean, before it was very tricky to culture them on a dish, and with this it's more like culturing cancer cells like HEC293 cells. So I'm going to show you some data first. This is about embryonic stem cells, and Sergey was really, I mean, he's actually one of the founders of BioLamin as well. So this article was a long time ago, but it just illustrates how these work. And basically, before, when you cultured embryonic stem cells on EHS extract, which is basically major gel tracks, you culture them as clumps, and they're actually in 3D as well. But when you culture them on laminins, you can split them as single cells, then they start making these kind of islets, and eventually the cells become fully confluent. And normally before, let's say these products, when the cells get too confluent, like here, you can see that here are blue cells, so these are not pluripotent anymore, but there are some cells there. So the cells start differentiating, but when you culture them on laminin 521, they don't really differentiate. This is vitronectin. You have the same issue here. This was published in Nature Communications 2014, and they had to do all these experiments. The karyotype of the cells are totally normal. They actually also did a SNP analysis to show in a more detailed fashion that the cells are totally normal. They show that they are pluripotent with a FACS analysis, and they also show that you can get basically all the 3 different germ layers, so that the cells are actually pluripotent. And this was done with about more than 20 different HPLC lines in the paper. Now that was 7 years ago, so now there's a lot more data in different publications, and we have actually cultured these embryonic stem cells for more than 200 passages at its best. This is actually, I mean, why does the laminin actually work that well? But this is actually a beautiful data from Leperle et al. in Madison, Wisconsin. And basically what they did was that they knocked out the alpha-5 chain in embryonic stem cells, and then they tried to culture them on different substances. And basically in all other substrates where they culture the cells died except for laminin 521. And basically what this article suggests is that the cells, when you plate them, the embryonic stem cells, if you plate them on vitronectin, they will actually somehow survive there until the cells produce their own laminin 521, and then they kind of grow on this laminin 521 that they produce endogenously. So this might be one of the reasons why this actually works like this. Malin Parmar, she's a professor in Lund working with neuronal cells and basically dopaminergic cells, and she she had developed a protocol using major gel, and basically the cells were cultured as EBs to make these dopaminergic cells, dopamine producing cells, and this is actually her protocol. So, and she was very interested in getting into the clinics, and since major gel is not really accepted in Europe, in clinical trials. So she was looking for something new, and basically she started using the laminins, and so she switched to laminin 111, and then what you could see is, I can't actually see that picture, but what you can see is that the cells look totally different in culture, and sometimes some people get afraid of this, but it actually worked really well in her case. The reason why she picked laminin 111 was that she could see that this was very much expressed in the in the area where dopaminergic cells were grown. So basically, her results was that she got 45 times increase in dopaminergic production rate, so which was really great. And so within 16 days, you could get material for over 500 patients in her case. And these cells were so pure that she didn't have to purify them in the end, which she had to do with her own method. And again, this is a clinically compliant way of doing them. Fredrik Lanner is a professor at Karolinska, and he has he's working with RPE cells. And I forgot to mention Malin is going to enter clinical trials with these cells now in January or December or January. And so Fredrik is working with RPE cells, and he also started using the laminins. And basically, first he used only one laminin, laminin 521 in his process. But when he did that, he had to manually kind of dissect these cells when they got pigmented to cut out the parts that were not pigmented. But then he continued his work, and he used a mix of different laminins there, and he could see that you can actually split these with trypsin and so on. So you don't need to do the manual dissection anymore. And now he has actually done quite a lot of animal studies, and right now he's actually producing his first clinical trial batch, and he also aims to be in clinical trials early next year. Lynn Yap, she was working with cardiac cells in Singapore, and then started working with the laminins and realized that these work really, really well compared to what she had done before. Let's see, how much time do I have? I don't have too much time. So basically, she showed the same thing. She could see that she used the biorelevant laminin 221 in her differentiation method. After that, she could see that in the single cell analysis that the cells are very, very similar in two different cell lines, which is very unheard of. And then the method became highly repeatable and clinically compliant. And she's also looking to get into clinical trials, but she's a bit more farther away. The results is what she has. She has the cells actually get vascularized when you inject them into the heart as well. I have to skip this, but basically ES and IPS. So most of our customers are working with ES or IPS cells, and there's not that many of these trials around the world, but there's maybe about 10 of them, and 5 of them are actually using the laminins. Malin Parmar and Fredrik Lanner, they're working together with Novo, and so there will be 7 trials using our products in a couple of months. And why do they use it? It's really because the process becomes so much more simple from the regulatory perspective and from the analysis perspective, because you can get to the endpoint, to the same endpoint more easily. Many times also you can actually get more cells when you use our products. The cells divide faster and so on. And then also that we can provide products that can be used in cell therapy trials. So originally we started from the laminin, we call the LN laminin, and this came from the academic lab. We didn't know any of the history of this cell line and so on. So when we started working with customers that wanted to come to the clinical trials, so we actually had to redo everything. So we started a new cell line. We made the vectors, everything new, so that we can have the whole history. And then we created these two new laminin lines where basically they're exactly the same, but here we do more testing so that the customers can use that in their clinical trial application. So there's much more documentation in this CTG product. And about these regulatory agencies, so most people, when they're doing clinical trials, they're aiming to get them approved in Japan, USA, and in Europe, and they actually all have slightly different requirements, but many people are trying to kind of, when you're developing a drug, you want to kind of do something that fits all of them. They're not that far away from each other. And then finally to this 3D culture system. This is mostly done by Alessandro Fiorentano, also in Malin Parmar's lab in Lund. Basically what we have done, so the hydrogels, the major gel and the hydrogels are basically gels, and the cells are growing inside the gel, but the biosilk is basically a silk fibers, and then where you attach the laminin to get the biospecificity for that, and so they're kind of in media, not in a gel. And basically many times with organoids, the problem is that the inner part of the organoid is different from, it behaves differently from the outside of the organoid, and this is something that they started working with, and they created a method how to make this in biosilk. And what they noticed was that in the outer part, when you have a gel-based organoid, there's spontaneous firing and action potential. Same thing with the biosilk, but the inner part with a gel-based is basically dead. It's not really an active neuronal cell. But when you culture that in this biosilk, you can actually see activity inside. And I can't show you more data about this because it's unpublished data, but it's accepted in Nature Communications. It will come out within a week or two, actually. Yeah, so summary, I hope that I was able to show you the 2D culture and how this has been used to actually get into cell therapy trials and why it has been important for the cell therapy trials. And basically, if you can make better cells for cell therapy trials, you could probably make much better cells for drug discovery, either using the 2D culture system or the 3D culture system. Yeah, I think I'm done. The time is up. Thank you for listening.
Thank you, Christian. I have one, well, it's a 3-in-1 question I have here for you. The first part of the question is, how come you don't need to add fibronectin or collagen in the gel to optimize your laminin-based BMEs? Or the cells producing these molecules themselves, or yeah, how does it work?
We don't know. I mean, and if you want to make a really, I mean, so there's two ways. You can try to make a very complex system that works, or you can try to make a simple system if it works. And it doesn't seem that the fibronectin or the collagen is actually needed, but we do know that these cells produce their own laminins. They might actually produce their own collagen and fibronectin and other extracellular proteins as well, but we actually don't know that. We haven't looked at it in detail.
Okay. Then with respect to clinical trials, do you need any special ethical approval for these clinical trials because of using these laminins?
So the laminins are, let's see, what was the name of it? I mean, we need to provide documentation to the, so we don't do clinical trials ourselves, but the customers that are using our products in clinical trials, they have to show how these products are made. So we have to send them quite a lot of paperwork, which they themselves send to the regulatory agencies. We are planning also to provide a so-called DMF, which is a drug master file in the US, which basically would mean that the regulatory authorities already are saying okay to our product, and so we don't need to send them to our customers anymore. But this is something that we haven't done yet.
Okay, Then there is a question from Kathy Mary. Can you comment on the glycosylation of your laminins? Do you know how human similar this is?
We haven't looked at it in detail, but they're very much glycosylated. Yes, they are. So these proteins have to be made in human cells, otherwise they won't work. We tried actually CHO cells as well in the beginning, and it didn't work.
Okay, then I think I have no further questions. And maybe one question from me. When you had your product and you wanted to have it accepted by the regulatory authorities, what were your major challenges?
Well, so basically, it cost quite a lot of money. So basically, we just needed to start from scratch eventually. I mean, so when we looked at kind of the history, I mean, the cell line was bought from ATCC, and it had been in the lab for a long time. And then also these vectors that were used to transfect the protein. I mean, we sequenced them, and they, but we didn't really know we couldn't really, we thought that it's easier to actually start from scratch where all kind of the components are known and the environment is known, because basically the differences between the FDA, EMA, and PMDA in general is the FDA doesn't really, they don't really care if you have animal products in the process. The Europeans care, and the Japanese care even more. And so there's these kind of words that you use, like animal free to the primary level or secondary level, and so on. So we needed to get rid of also BSA in our manufacturing process as well. So that was also the thing. So but I mean, it took us actually, I think 4 or 5 years to make this cell line, because we bumped into all these kind of problems when we were taking away, let's say, BSA, and we changed the media, and we have to make, and so we also had to make a purification method that was totally different from the first one, because the first one is not also scalable in a way. So it took a long time. I mean, we have one person that was working on this full time together with partners for 5 years.
Yeah, it's a challenge, I can imagine.
And then we had to, of course, have contacts with the regulatory agencies so that we understand that part. So we had to learn everything from scratch.
Okay, then a final question, I think. Yeah, we have just time. How certain can you be that the laminin pure BME does not generate stem cells with an identity that could be more tumorigenic? Has there been preclinical studies that verify whether there is no increased risk in these grafts when compared to Matrigel?
So we haven't looked, I mean, so yeah, that's one of the obvious questions when you see that the cells grow much faster. And actually that's, I mean, when we have been showing this to potential customers, that's was something that they look at and they think that looks, might look bad at least. But it doesn't, I mean, we haven't done that, but what we can see from, and the reason why our customers use this in clinical trials is that the process becomes more robust. It's more predictable. You get the right results. So it's not that they, I mean, that's really the reason why they use it. So I would say that kind of talks against them being more tumorigenic.
Okay, then I do not have anything more. And with your presentation, you also addressed one of the previous questions regarding tissue specificity. So I wasn't aware there were so many different laminins, each targeting very specific cell types. So I learned something here. Yeah.
Sometimes combinations as well. So
okay,
making it even more complex.
It's almost indefinite. Okay, then I think we are done with this webinar. So unless the NC3R organizers want to say something, I I close this this meeting. Emma, are you?
Yeah, we'd just like to say thank you to everybody who has attended. We'll be placing a recording of the session on the NC3R's website early next week. It's the same link that you used to register. So if anybody would like to watch it again or to share with colleagues, it will be there from early next week.
Okay, that was the final touch. So thank you everyone, thank you to the speakers for taking the time to present their work, and thank you for the participants to be with us, and hopefully we all learned a lot of things about BMEs we didn't know before. Have a nice day and stay safe.